Hi there, my name is Aurelia Reynolds, and welcome to my presentation, The Effects of the LON Protease on Topoisomerase 4 Activity in Caulobacter Crescentus. So first I want to introduce Caulobacter Crescentus. It's a gram-negative alpha protobacteria that lives within freshwater environments. The strain that we'll be using for this study is the NA1000 or the CB15N. So a couple things about Caulobacter is that they have an unusual cell cycle. We're going to go further into detail of this, but this diagram provides a quick insight into the process. In Caulobacter, there's a protein called the LON protease, which is responsible for the degradation and regulation of many different proteins. And through transposon and RNA sequencing techniques, we're allowed to say and label certain genes in Caulobacter as essential to the cell's vitality, as in that these genes are necessary for the cell to live. A particular knockout called the delta LON, these cells lack a production of the LON protease. In delta LON mutants, we're known to have some phenotypes, including increased levels of DNA replication, altered cell cycles, and longer filamented cells. So looking back at the cell cycle, the Caulobacter has two different stages, the stalk cell and the swarmer cell. The swarmer cell differentiates into the stalk cell, which immediately then enters the S phase. This initiates DNA replication and completes the cell cycle process. The swarmer cell then continues to stay dormant in the G1 phase until it differentiates. This is called asymmetrical replication. It also should be known that within the cell cycle, there are two different redox states that the cell is in, a reduced state, which is seen in G1 and G2, and an oxidized state, which is seen during the S phase, which is during DNA replication. Now, these different types of redox rates of the cells are important for the regulation of a lot of different proteins within DNA replication. Within DNA replication itself, there are four major regulators: CTRA, GCRA, CCRM, and DNAAA. Out of these four, two are what affect our replication the most, which are CTRA and DNAAA. DNAAA is responsible for the initiation of DNA replication. Once it's phosphorylated by HDAA, it is able to bind to the site of replication and open up the double helix in order to recruit the other DNA replication factors. This diagram shows a proposed model that includes the recruitment of topo4. So topo4 is a very important protein during DNA replication. It has two main functions, one being the relaxation of supercoils and the other one to decanate bacterial chromosomes. So when DNA replication happens, the DNA replication complex binds the DNA and opens up that double-stranded helical structure. In this case, what is called a replication fork. And at the end of that replication fork, before the DNA replication complex, it actually creates over-twisting and tension. Think of this as twisting an old telephone coil that's already twisted. It just twisted within itself, creating almost knots. So in order to alleviate the tension so that the DNA doesn't break, topo1, topo4, and another protein called gyrase have to go through and clip that DNA and rebind it so it alleviates those supercoils. Another function of topo4 is the decantation property. This is specifically within bacteria. So after bacterial chromosomes are replicated, they're actually linked together in a chain-like function. And what topo4 goes and does is it before cell division, it goes and it clips one of those catenated chromosomes, it unlinks them, and then it fuses the DNA back together. So it has two separate bacterial chromosomes, which are then able to separate into the two daughter cells. Topo4 has two different subunits, part C and part E. Part C is responsible for the mechanistic activity of topo4, and part E is responsible for the catalytic activity. Topo4 is active when part C and part E are bound. However, part C is able to bind to DNA alone during replication for the relaxation of these supercoils. And the decantation activity happens when part C and part E bind together at the end of DNA replication. It should be noted that within this presentation and the study, topo4, part C, and part E might be referred to topo4A, subunit A, and subunit B, respectively. So two of the proteins that are really important in topoisomerase 4 activity are NSDA and GAP-R. GAP-R is a nucleotide-associated protein, which is a protein that specifically binds to certain shapes and not a particular DNA sequence. This is important because GAP-R actually binds the shape of supercoiled DNA and recruits topo4 for those relaxation of those supercoils. NSTA affects the decantation activity. It actually inhibits by binding to the par C subunit and not allowing the topo4 to unbind those chromosomes. So what's really important is what we're seeing in the RNA sequencing and transposon sequencing data. So in an RNA sequencing data, you're actually allowed to see the different levels of RNA expression within different genes, within the wild type and the delta LON mutant. This is important because if there's an upregulation or more production of a certain protein, you're going to be able to see it in higher levels of RNA expression. Or if something's downregulated or it doesn't produce enough proteins within one of the strains, you're able to see this in lesser RNA function. With transposon sequencing data, it basically measures the amount of transposon insertions, which you can see right here. And those insertions actually interrupt the gene. And when you have too many of them, the gene is not able to reproduce in a certain strain. And what's really important is that this is allowed to show what genes are allowed to be knocked out in certain different strands. So here we're actually able to see that NSTA has no insertions in either a delta lon or a wild type. And this is very important because this is saying that NSTA should be labeled as an essential gene in a delta lon mutant. We're also seeing that there might be a slight upregulation of that RNA sequencing data, as in there's more expression in a delta lon mutant, and we don't really know why. We have increased levels of DNA replication activity, so you would think that you would have increased levels of supercoiling, increased levels of decatenation needed, and therefore increased levels of topo4 needed, but instead NSTA is showing that it's increasing and actually inhibiting topo4. On the other hand, when we're looking at GAP-R, we're actually seeing, somewhat of, again, another upregulation of RNA expression. We really don't know. We have to look at some statistical data for that. But more importantly, we're seeing insertions, meaning that GAP-R could not be produced in a delta lon, saying that the activation property of topo4 is not needed in a delta lon cell, which is pretty interesting. So what are some of the questions that we're trying to ask? We're asking, how is topo4 activity regulated in Caulobacter Crescentus? We also want to know, how is that regulated in specifically a delta lon mutant? And is what we're seeing a direct result of a lacking of lon protease, or is it due to downstream effects? As in, does the lon protease directly affect, does it directly interact with topo4, NSTA, or GAP-R, or is it something else? We're also trying to look at what other genes are important in this topo4 regulation. And are those genes upregulated and downregulated in the same fashion as NSTA and GAP-R, as in, are the inhibitory proteins upregulated and the activation proteins downregulated? Ultimately, because we want to look at how does lon affect the topoisomerase 4 activity in Caulobacter Crescentus. I do have a quick hypothesis, and that is genes that are involved with topoisomerase 4 activation will be downregulated in a delta lon mutant, while genes involved with topoisomerase 4 inhibition will be upregulated in a delta lon mutant. I'm thinking that some of the phenotypes that we're seeing in these delta lon mutants are a result of these inactivation and activation, as in the longer filamented cells in the abnormal cell cycle. Why are we asking these questions? Well, it's very important. Antibiotics. Topoisomerase 4 is a very common target for a lot of antibiotics out there. And while Caulobacter Crescentus is not a harmful bacteria, it is important to study the topoisomerase 4 so we can apply it to other types of bacteria and hopefully help with the study of antibiotics in general. Antibiotics resistance is a very prevalent topic. It's becoming more and more relevant in a lot of different diseases. And so any type of study that helps, with antibiotics in general, antibiotic resistance is beneficial. So some of the methods that I'm looking at the statistical analysis of the raw data. I'm able to look at the upregulation and the downregulation in different folds of log. I'm also able to see the p-values and the false discovery rates, also known as FDR, through this data. I'm using what's called an integrated genome viewer. This is where, again, we're able to see the RNA sequencing and the transposon sequencing data. And here's actually a better view of those insertions. So you can see that in a lot of different genes, there's a lot of levels of insertions in a delta lon versus a mutant. And we're also able to see that different type of expression of RNA. I'm also using RStudio just to kind of look at the different upregulated and downregulated genes and how they're statistically significant within volcano plots. I'm also using this thing called the string networks, where I'm able to put in different types of genes and see the different types of enrichment within the pathways. I'm also able to see how genes are related. So here we're able to see PAR-C and able to see all the different types of proteins and genes that are related to PAR-C through different types of relations, experimentally, that kind of stuff. And lastly, I'm doing a larger literature review just because I really want to be able to understand the entirety of topoisomerase 4, DNA replication, all that kind of stuff in Caulobacter. So looking here at the statistical data of NSTA and GAP-R, we're able to see that the top here is the transposon sequencing data and the bottom is the RNA sequencing data. And we're actually able to see a different type of fold difference within the GAP-R and the NSTA. So here we're saying GAP-R is, has more insertions and NSTA has less. And actually these values are significant. They both have p-values of less than 0.05, and they also both have false discovery rates of less than 0.05, which is very important because those values are significant now. Looking at the topoisomerase subunits, subunit B and subunit A, which are also known as Part E and Part C. we're able to see that there are no insertions in either the wild type or the delta lon strain. This is also very important. Unfortunately, again, we're showing the transposon sequencing, the RNA sequencing. There are no values that really are any significance. The log values show that there's not a high enough fold to really say that there's a difference. The p-value and the FDR actually say the same thing, that those types of values don't have any statistical significance. Looking at the DNA gyrase, which is a protein a lot like topo4 in the way that it functions with the supercoiling, we're actually able to see this again. The insertions are seen in neither the delta lon or the wild type, saying that this gene is very important for the vitality of a Caulobacter cell. Again, the transposon sequencing and the RNA sequencing data, those values, the folds are not large enough for us to see a difference. Here we're able to see a volcano plot with upregulation of some of the genes that we've been looking for. They are statistically significant just because of the fact that we have them above the line. And we have a lot of other genes also within that area that I want to look at further. So what do we know right now? We know that NSTI and GABAR are definitely affected by LON in some way, we just don't know how. We know that there are numerous other genes involved with DNA replication that are also affected by LON. And also in a study done by Zian et al., we also know that LON actually degrades DNA-bound proteins. And this is really important in some of the things that I want to propose going forward. So what is proposed? Well, I think that topoisomerase part C and part E are conserved genes, that they have to be in both that wild type and delta lon mutant in order for the cell to be viable. I also say the same thing for DNA gyrase. Both of the subunits A and B also are conserved. They need to be there in order for the cell to live. And lastly, I think that the insertions from GAPR are due to it being to a DNA-bound protein. This I have to investigate a little bit further. Obviously, my results are not concluded at this time, but that's where the further directions come in. I have a list already of a lot of genes involved in DNA replication and topoisomerase 4 activity, but I want to investigate that further. I really want to go into this volcano plot and really see what types of genes, if those are DNA-bound proteins, if they're just DNA replication genes in general, what kind of genes they are. I want to see those trends, and hopefully I'll be able to conclude those within the next few weeks. I want to give special thanks to Dr. Peter Chan for providing a really great environment for me to work in the past 2 semesters. He took me to my lab with COVID. It's been really difficult, but he's provided so much encouragement and so much guidance within the past year. I'd like to thank Dr. Auerbach and Dr. Furman for their endless support over the past 4 years without the ICONS program. They've been really incredible mentors, really incredible professors. Julia Mendel, she's been a fantastic peer. She's provided a lot of support and feedback on this project, and obviously my family for always supporting me throughout my time at UMass. For any questions, you are always able to email me at AureliaReynolds at umass.edu. I'm also holding a question session on April 26th from 6 to 7 p.m. via Zoom. If you would like to attend, please just email for an RSVP, and I will be happy to provide a Zoom link. Thank you.